In this chapter, we're going to look at different types of mutations that occur in DNA and how they are repaired. In the first section of this chapter, we're going to see what mutations are. Mutations are changes in genetic information that are heritable, which means it can be passed on to progeny. Mutations are the sources of all types of genetic variation that we see today. It is because of mutations that organisms have evolved to form new species and different types of organisms. Mutations are very commonly used to understand the function of proteins and other aspects of living organisms. When we look at the types of mutations in multicellular organisms, we should remember about two types of mutations. Somatic mutations are mutations that occur in the somatic cells that are basically cells that do not have any role in reproduction. Somatic cells do not give rise to gametes. Germ-line mutations are mutations that occur in germ-line cells, and germ-line cells are the cells that give rise to gametes. Hence, germ-line mutations are the ones that are passed on to offsprings, while somatic mutations remain within the organism and is not passed on to the offsprings. Let us look at an example of somatic versus germ-line mutations in a fish. In the case of a somatic mutation, there's been a change in the DNA in one of the somatic cells as shown in the box. When this cell undergoes cell division, the mutation is passed on to the daughter cells, and hence as the fish grows through the process of mitosis, we have a population of cells within the fish that harbor that mutation, and hence we have a collection of mutant cells. These cells are not involved in the formation of gametes and remain within that fish. Let's look at what happens when you have a germ-line mutation. In this case, a mutation or a change in the genetic information has occurred in a germ-line cell. When this germ-line cell makes gametes, then the gametes harbor this mutation. Since the gametes harbor the mutation, when the fish undergoes sexual reproduction, depending on the kind of mutation, either all the gametes or in this case, some of the gametes have the mutation, and hence we have some progeny that will harbor the mutation in every cell of theirs, and hence will be a mutant fish. While we will have other progeny where they do not carry the mutation because the gametes do not carry the mutation. Thus, somatic mutations are not passed on to the next generation of progeny, but stay within the cells of the organism as they undergo mitosis, while germ-line mutations can be passed on to the progeny via the gametes. In this section, we're going to look at the cause of mutations. Now, mutations can arise spontaneously or can be generated through the action of chemicals. Spontaneous mutations usually arise due to natural changes in DNA structure or from errors in DNA replication. Induced mutations, on the other hand, arise due to environmental agents like chemicals or radiation. We're going to first look at spontaneous mutations. In the following section, we're going to look at how DNA can be repaired. Now, as we've seen that in spite of constant exposure to radiation or mutagens or any other agents that can cause mutations, the rate of mutations in DNA is actually very low. And the reason for that is that cells have ways to repair DNA. In fact, cells have multiple repair systems to repair any kind of damaged DNA. And the availability of multiple repair systems gives rise to redundancy, and this redundancy is critical for the survival of a cell because it has to ensure that the integrity of its DNA is not compromised, since it is the genetic material and has all the instructions for the survival of a cell. The different types of repair mechanisms that are available are mismatch repair, base-excision repair, nucleotide-excision repair, homologous recombination, and others. In the following slides, we're going to look at a few types of repair mechanisms. The first repair mechanism we're going to look at is the mismatch repair. Now, during DNA replication, a mismatched base is added and is not repaired by DNA polymerase's exonuclease activity, then that particular mismatched base is usually repaired by the mismatch repair mechanism. So let's look at the figure where we can see that there is a mismatched base. This mismatched base is identified by the mismatch repair complex, which is a complex of proteins. In order to do the repair, the mismatch repair complex must first identify which is the new strand and which is the old strand. In order to determine which is the new strand and which is the old strand in the case of bacteria, what the complex does is it looks for a site in the vicinity of the mismatch which has the sequence GATC as shown in the next box. This GATC site is usually methylated in the case of bacterial DNA. When DNA synthesis occurs, the methylation of the GATC site is delayed, and hence if the mismatch repair is able to recognize the mismatch base that's present in the DNA before the GATC site is methylated, it can definitely identify the new DNA from the old template DNA. Once it recognizes the new strand from the old strand, it loops the DNA, and this way it is able to determine where the new strand is and which base it needs to repair. The next step that it does is it will cut the new DNA strand as shown in that box, and that new DNA strand, a portion of it, is actually removed. Thus, right now, we can see that there is a stretch of DNA that is only single-stranded. This stretch of DNA does not have the mismatched base anymore. DNA polymerase then comes in and fills up the gap and puts in the correct nucleotide in place, and this way the mismatch repair is repaired and we have proper DNA again. Another type of DNA repair is direct repair. Unlike mismatch repair, direct repair will correct bases that are chemically modified by returning them back to their original structure. There's no removal of the bases or breaks in DNA strands in order to correct the modified base. Let's look at the example as shown in the figure where a guanine has been modified by the addition of a methyl group to the oxygen at the 6th position. And thus we have O6-methylguanine. And in order to retain the structure or get back the original structure of guanine, that methyl group has to be removed. An enzyme called methyltransferase does the job of removing the methyl group, and now it restores the guanine to its original structure. Thus, there's no need to remove the modified guanine from the DNA strand, and hence direct repair directly corrects any kind of chemical modifications that might be there on the nitrogenous basis. Another example of direct repair is when thymine dimers are formed due to UV light. So in this figure, we can see that thymine dimers have been formed because of the exposure of DNA to the UV light, causing a distortion in the DNA structure. These thymine dimers can be removed by an enzyme called photolyase, which will recognize the thymine dimers and bind to the DNA where the thymine dimers are present. Once the thymine dimers are identified and photolyase binds to them, it can remove the bonds that form the thymine dimer in the first place and restore the DNA structure back to its original form. Another type of repair mechanism is the base-excision repair. If there is a situation where a nitrogenous base is damaged, it can be corrected by base-excision repair. So here is an example where we have a damaged base, right, shown in the black box. This damaged base is first removed by the enzyme DNA glycosylase, and the removal of that nitrogenous base from the DNA structure gives rise to an AP site. AP stands for apurinic if the nitrogenous base was a purine, or apyrimidinic if the nitrogenous base was a pyrimidine. Once the AP site is generated, an enzyme called AP endonuclease cleaves the phosphodiester bond at the 5' end of the AP site. So if we look at the picture, AP endonuclease recognizes the AP site and it removes the ribose sugar and the phosphate bond from that position, and now we have a break in the DNA where we have a missing nucleotide. This break or gap that's now generated can be filled in by DNA polymerase, and then a new nucleotide that is complementary to the other strand is added, and thus the base excision repair is done. Finally, DNA ligase will come to form the final bond, and we get new DNA without any mutations. Another type of repair mechanism is the nucleotide-excision repair. This repair system looks for distortions in the DNA structure that can be caused by mutations like pyrimidine dimers. Here in the figure, the black box shows a part of the DNA that it's showing a distortion due to a mutation. The distortion in the DNA is recognized by a complex of proteins and enzymes as shown in the figure in the black box. Once this complex of proteins and enzymes recognizes the distortion in the DNA, what it does is it separates the DNA strand so that the DNA is no longer double-stranded and keeps the DNA strand single-stranded by recruiting single-strand binding proteins. Once the DNA is single-stranded, the DNA strand that has the mutation is cleaved on two ends and it is removed, thereby forming a big gap where you have a huge stretch of single-stranded DNA. The gap is filled by DNA polymerase, and DNA ligase will add the final phosphodiester bond, thereby repairing the DNA, and hence now there are no distortions in the repaired DNA. All these examples that we have seen so far are when we have minor repairs in the DNA. Let's look at what happens when there are double strand breaks in the DNA. A lot of times, DNA can endure double strand breaks due to ionizing radiations and even UV radiations. If double strand DNA breaks are present, then the preferred choice of repair is homologous recombination. This homologous recombination is similar to the mechanism of homologous recombination that we learned during the DNA recombination section a few chapters ago. The difference in this case is the recombination is not occurring during meiosis. So let's see what actually happens. So when the DNA has undergone DNA replication, then the homologous recombination is usually used. In the replicated DNA, you'll have a sister chromatid and you'll have the other sister chromatid where the double-strand DNA break is present and hence it is damaged. The information that is present on the sister chromatid, since the DNA is replicated, will be used to repair the DNA double-stranded breaks. And the way that will happen is similar to homologous recombination. The double-strand break that's present on the DNA will be cleaved a little more so that you end up with these uneven ends. One of the ends will invade into the sister chromatid and branch migration will occur. Finally, we will have DNA polymerase that will start synthesizing new DNA on the broken DNA. And in the end, we will end up with two holiday junctions. These holiday junctions will be resolved and we will have our double-strand breaks that have been repaired. Another way double-strand breaks in DNA are repaired are through non-homologous end joining. This is usually the preferred method when the cell is in the G1 phase and hence there is no sister chromatid available in order to help serve as a template to rectify the double-stranded break. In non-homologous end joining, proteins recognize the broken ends of DNA and they directly join them as they are. The problem with this kind of end joining is that it can give rise to errors like additions, deletions, or translocations. In the figure shown to the right, in the boxes, we have the proteins that have recognized the double-stranded breaks in the DNA, and they will simply join them as is. It is not a foolproof method, but at least you won't have broken DNA. With this, we come to an end of the various repair mechanisms that are present in the cell, and this table summarizes the different methods that we looked at. It's important that cells have good DNA repair mechanisms because in the event that the repair mechanisms are not able to perform their function, there are many diseases that arise. This table summarizes different diseases that arise due to defects in different DNA repair mechanisms. Thus, it's absolutely important for a cell to be able to repair its DNA in the event of any kind of DNA damage. With this, we come to the end of this chapter. Please ensure that you come and bring your questions when we meet again in the next class.